advisement of Professor Iwai and Professor Lüthi at the University of California, Berkeley. And so today, I'm going to be discussing a simple technique that we use to trap and release microbeads from a dynamic microarray. And so today, I'm going to be first discussing why we want to be able to trap and release microbeads in an array. Then, I'm going to be discussing our technique to do so. And lastly, I'm going to be discussing some future directions for this project. So for chemical and biological applications, there are essentially two main types of microarrays, static and dynamic microarrays. So static microarrays refer to chemicals or biomolecules which are mobilized onto a static substrate. In contrast, dynamic microarrays consist of chemicals or biomolecules which are mobilized onto mobile substrates. And this consists of either microbeads or microparticles or cells. But between these two technologies, dynamic microarrays present a superior technique. And this is due to a number of advantages. In particular, the faster diffusion times, the lower required reagent volumes, and the higher surface to volume ratios associated with microparticles, and more specifically to microbeads. But the critical barrier to entry for this technology is really in terms of particle handling. And so the goal of this project here is to improve particle handling. And we do this through two distinct fronts. The first of which is by increasing the particle trapping density. And so our personal motivation for this actually originated back at MEMS 2009. And there we presented a technique where we detected special types of DNA on the surface of microbeads. And the way that this works is that if the DNA that we're looking for is present, then we'll be able to detect a certain level of fluorescence on the surface of microbeads. However, if that DNA is not present, then the fluorescence response is reduced. What we ended up finding was that if our sample size was not high enough for an experiment, then we could not conclusively determine whether or not the DNA was present. And as a result, we had to run multiple experiments in order to be able to get a clear view of whether or not the DNA was present. However, by increasing the particle trapping density, we can greatly increase the sample size for a given experiment. And not only can we achieve statistically significant results for a single experiment, we can also begin multiplexing reactions on a device. And we can do all of this without needing to run additional experiments or having to increase the size of our microdevice. So the next aspect that we focused on was achieving microarray resettability. And so this issue actually really arose from our specific process of running bead-based experiments. And so the way that this works is that first we take our microfluidic device, then we can run one single bead-based experiment, and then we can take that device and throw it away. Then if we want to run additional experiments, we have to spend the time and the money to fabricate multiple devices corresponding to every single bead-based experiment we want to run. And for us specifically, this ended up coming down to roughly 70% of our time was devoted to fabricating devices, and 95% of our money. And so then you finally get back to square one and you have your new devices. But the reason for microarray resettability is really to streamline this process. And instead, simply reset the device by releasing all the microbeads from the array. And by doing so, you can run multiple bead-based experiments using a single microfluidic device. And so previously, there are two primary hydrodynamic microparticle arraying technologies that I'll be discussing today. The first of which was developed by Professor DiCarlo of UCLA a long, long time ago when he was actually a member of Lüthi lab at UC Berkeley. And for this, he uses elevated U-shaped weir structures in order to trap cells. As you can see, this technique was quite effective. But the issue here is that there's certainly room for improvement in terms of the overall trapping yield, specifically one bead or one cell per trap. The following year, a group at the University of Tokyo, specifically Professor Takeuchi's group, came out with a technique where they regulated two distinct fluidic resistances in order to control microbead movement. By doing so, they they were able to achieve a very high efficiency technique to trap an array of microbeads. But the issue here is not really in terms of trapping, it's in terms of the microarray resettability. And following this, they put forth a number of papers in order to achieve resettability for this technique. The first of which is to actually plant aluminum plates underneath the traps and then shine a laser to create a microbubble to physically displace the beads from the traps. And while this is extremely effective if you want to release one single bead at a time, it's horribly inefficient if you're looking to release a high number of microbeads to rapidly reset a microarray. This next example was actually presented by one of our co-authors, Iwai. And here what he did was actually use the accurate placement of microfeatures in order to facilitate the release of microbeads during reverse flow. And while this was able to achieve microarray resettability, as a result, it actually limited the overall trapping density. So the following year, at MEMS 2009, Kosuke presented another technique where he used pneumatic valves in order to facilitate the release of microbeads. But for us, what we were looking for is really the easiest, most simple technique to be able to trap and release microbeads, and specifically to achieve both high-density trapping as well as microarray resettability simultaneously.